Hello, everyone. Welcome back to our week 4 of the OSSA Ontario Science Students Association's lecture series. Today we have an amazing guest speaker, Dr. Russell Bishop. Dr. Russell Bishop was actually a professor of mine during Biochemistry 4N03. Amazing course. I highly recommend it. But also, Dr. Russell Bishop is also an amazing researcher at McMaster. Russell obtained his PhD in biochemistry at the University of Alberta, where he then went on to do research and to discover several bacterial membrane lipoproteins in the laboratory of Joel H. Weiner. During his PhD research, Russell spent 1 year in the Department of Biochemistry at the Max Planck Institute for Developmental Biology in Germany, where he studied in the laboratory of the late Joachim Volker Holte. And then Dr. Bishop's postdoctoral studies began with investigations of antimicrobial peptide biochemistry in the laboratories of Robert S. Hodges and Cyril M.K. in the Protein Engineering Network of Centers of Excellence at the University of Alberta. And then in 2000, Dr. Russell was recruited as an assistant professor for the Department of Laboratory Medicine and Pathology at the University of Toronto, where he held a cross appointment in the Department of Biochemistry. Then also receiving tenure in 2005, and then following was recruited as an associate professor to the Department of Biochemistry and Biomedical Sciences at McMaster University, where Dr. Russell Bishop has been there ever since, and then is also a member of the Michael G. DeGroote Institute for Infectious Disease Research at McMaster University, and has recently completed a 5-year term as an editorial board member for their Journal of Biological Chemistry. His research group continues to study mechanisms of gram-negative cell envelope biogenesis. Special thanks to Dr. Bishop. Quick before we start, if you have any questions, I'm sure Dr. Bishop would be happy to take them, whether it is during the lecture or at the end of the lecture. If you feel uncomfortable with unmuting yourself or showing your face, you can put them in the chat. I'll be reading them out at the end of it. Keep in mind this lecture is recorded, so anything you do ask will be recorded, so keep that in mind. But otherwise, I hope you enjoyed this lecture. I'll pass it on to you, Dr. Bishop.
Thanks, Israel. So yeah, I'll help you a little bit with the German. My experience at the Max Planck Institute in Tübingen was with a guy named Joachim Volker Holte. And Joachim it's interesting, the name in German, they pronounce every syllable. So that's how it's easier to figure out how to do it, how to pronounce that. So I should have given you a little help with that. That was really hard. Everybody struggles with his name. So I'm sorry to have set you up. Okay, so my purpose here is to address the Ontario Science Students Association. Is that my understanding? You didn't tell me much about what you guys were.
Yeah, so we are a association for Ontario science students. So all across Ontario, we have 9 member universities that are on our board, including the McMaster Science Society, along with other science societies such as in Western, Waterloo, Laurier, Carleton, University of Ottawa, University of Windsor, and then I always forget the additional ones, but in total we have 9 member universities who we try to service the science students all across there. But this time we're slightly more focusing on McMaster, and so most of our guest lectures for our lecture series, our inaugural lecture series, have been from McMaster, and then also a lot of students have also been from McMaster. But yeah,
well, that's okay. I'm looking forward to having a chance to share with you about the research in my lab. Of course, this is very different from a lecture. As you took my course Biochemistry 4N03, it's called Molecular Membrane Biology. We try to understand how membranes work biochemically. And in cells, we use mutations and genetics to understand if the phenotypes match what we see in vitro with purified components. And so the challenge is to understand the structure and function of membranes. And I'm going to talk about a specific enzyme that's a membrane protein. It sits in the outer membrane of E. coli, and it takes a palmitate chain. It's a 16 carbon saturated acyl chain, and it attaches it to a lipid in the outer membrane of E. coli called lipid A, also referred to as endotoxin, because it triggers an inflammatory response. And we believe that the enzyme does this to interact with the immune system of the host. So maybe by making the cell membrane more amenable to dealing with the attack that's laid out by the immune system, the bacteria use this kind of as a response mechanism. There's a number of different means by which adding a piece of spaghetti from a phospholipid to another lipid might manifest itself. And so there's quite a bit of possibilities that we haven't considered just in an opening statement like that. But maybe by the time I've finished explaining it to you, you'll see the full complexity. Perhaps the most valuable thing that came out of this research was we developed an adjuvant that's a part of a vaccine. And that vaccine was used to vaccinate teenage girls in Ontario and other parts of Canada when they're like 12 years old against the human papillomavirus. And it's been about 20 years now, and people are starting to see the results. And it shows that when the girls got vaccinated against the human papillomavirus, that cohort is no longer showing any sign of cervical cancer in their 20s. So it's the first insight that comes from it. And we had to be able to make a vaccine that had an adjuvant that would work, and understanding how the palmitate chain gets attached to lipid A was actually a fallout of that work. We didn't expect it. It was unanticipated, but it was just an interesting question. How does an enzyme transfer an acyl chain from a phospholipid to another lipid in a detergent micellar environment? And we ended up finding a way of actually preventing cancer. So it's like, you don't know when what you're doing is going to matter or not, but we were interested, and I'll try to give you a feeling for how that happens. Okay, so PagP sits in the membrane. Oh, here's the people that have primarily helped with this. We'll start at the bottom. On the left, there is Charneil Dixon, and then Sanchia Miller and Lisette Maldonado. They were the last 3 PhD students to go through my lab. And today, Mo Shariari is actually helping me make some videos that I've been using in this presentation today. And the individuals at the top here on the top left are Louis Kay, who is an NMR spectroscopist at the University of Toronto. And Peter Huang did his PhD with Louis and is now an MD PhD who has a clinical and research practice at the University of Alberta back in Edmonton, where I did my PhD. There's also a crystallographer named Gil Privé who helped us get crystal structure information from PagP. And Regio Pommes, who's from France, is a computational chemist. And he has, along with Chris Neal on the right here, they had done molecular dynamic simulations on PagP. And so between the group in my lab who are purifying the enzyme and doing site-directed mutagenesis and developing assays are interacting with the structural biologists who are crystallizing the protein and giving us biophysical pictures of how it might behave. And so it's a collaborative effort. It can't be done by any one individual. So PagP sits in the outer membrane of E. coli. This is a schematic diagram of what the cell envelope of E. coli might look like. It's like a cross section through the cell wall, and there's an inner membrane which has a phospholipid bilayer structure, characteristic of any biological membrane, really. But the bacterial cell envelope is different, unlike, say, the erythrocyte in your blood, where there's a cytoskeleton on the inside. The bacteria actually have a cell wall on the outside of the membrane. And attached to that is an outer membrane in E. coli, because it has 2 membrane systems. And the outer membrane system is very different because the external leaflet is made up of an unusual lipid called lipid A, or endotoxin. And it can be highly decorated with sugars, as shown here schematically. And the proteins that are embedded in the outer membrane are structurally distinct. They have beta barrel structures as opposed to transmembrane alpha helical structures. We'll see some examples more close up as we go. And the outer membrane is asymmetric because it has two different kinds of phospholipids on either side, the glycerophospholipids like those in the inner membrane and the lipid A in the external leaflet. And so understanding how the bacteria structure their cell wall gives us an idea of how to introduce antibiotics so that we can be able to treat infections. So it's one of the $64,000 questions to understand how the permeability of the outer membrane is maintained and regulated. So the enzyme that we're looking at that sits in the outer membrane is a small beta barrel protein, catalyzes this chemical reaction. It takes a palmitate chain, which is a 16 carbon saturated fatty acid, which is esterified to the backbone of a phospholipid known as phosphatidylethanolamin. And there's two acyl chains in every glycerophospholipid because the glycerol moiety has three hydroxyl groups. One is phosphorylated and then the other two get esterified with fatty acyl chains. And so it's actually the SN1 chain that gets transferred and it gets attached to this hydroxyl group here at position 2. Are you guys able to see my cursor on the screen or do I have to?
Yep, we're able to see it.
You're able
perfectly.
to see it. That's good. Thanks. So the transfer of the acyl chain to this hydroxyl group creates a lipid A that's been modified by the addition of this palmitate chain. And that actually prevents or changes the way the lipid interacts with our immune system. There are receptors of immune cells that bind to this. And once it's been modified, it attenuates the inflammatory response and sort of helps the bacteria to fight against the immune system. But what we have is a very simple enzymatic reaction where there's two substrates and two products. And the simplest mechanism that can describe it is not either of the two that are shown here. These are the more complicated mechanisms that actually can describe PagP, and we've only learned about this in the last year or so. So PagP, using two substrates and two products, has a kinetic profile that would involve the binding of the first two substrates and then the release of the first two products. And so there's a ternary complex where the enzyme binds to both of its substrates, the palmitoylated phospholipid and the unpalmitoylated lipid A. And so it forms a ternary complex between the enzyme and the two substrates, and then a reaction will proceed to produce the products. And what's happening is there's going to be an isomerization of the enzyme. So it goes from one ground state structure, which is able to bind the phospholipid substrate. And if it finds the right phospholipid acyl chain, if say, for example, there's a myristate chain, which is 2 carbons shorter at this SN1 position, the enzyme will then release that substrate because it's the wrong one. And if it gets the right one, which has 16 carbons, that will trigger a conformational change in the enzyme that allows it to bind its second substrate. So it's part of the mechanism by which the enzyme selects the correct acyl chain. And it's only in the isomerization to this excited state after the correct substrate binds can we deduce the catalytic mechanism. And we've only just done that very recently. So that's the one thing I really wanted to talk to you about today. So there are two potential iso mechanisms that can explain PagP: iso ordered by, and iso Thorell chance. And I think it's the latter one here. The equations that describe this have 14 terms in the denominator. They're more complicated than the typical one that has only 11 terms. This was discovered by Mo Cleland back in 1962 as the equation that describes a two substrate, two product enzyme called an ordered by mechanism. Well, what we have here with PagP is actually an iso-Thorell chance mechanism, where instead of just the first substrate binding and the second substrate binding to form a ternary complex, the ternary complex resolves itself so rapidly that it effectively has no lifetime. And so we use an approximation in what's called Thorell chance, named after the Nobel laureate Hugo Thorell and Britton Chance, who invented the spectrophotometer. They described this as an aspect of a dehydrogenase mechanism back in the 1950s. And we now know that an isotherel chance mechanism is in fact geometrically or like algebraically, it's identical to ordered by. And it's one of those dirty little secrets of dehydrogenase kinetics that it's been known for many years that you can't really distinguish between these two mechanisms using enzyme kinetics because they're described by the same algebraic equation. The meaning of the constants in these two mechanisms are different. So the individual rate constants that make up equilibrium constants have different forms, but the way they're represented algebraically, they are in fact symmetrical or identical. So they're complementary mechanisms. The thing is that if you have an iso mechanism, sometimes it can do things that an ordered by mechanism won't do. So let me just show you initially how we figured this out. So this is a little simulation of PagP in a detergent micellar environment where it's exchanging with all kinds of lipids that are added to the system. And we showed that as we increase the lipid concentration, we could get sort of a gradual increase in activity, but it only worked at a particular detergent concentration. And once we found a detergent concentration that would support an increasing one-to-one relationship with added substrate, we could see that it was behaving in a hyperbolic fashion, kind of like Michaelis-Menten kinetics is expected. And if we diluted the system with fresh substrate, it would take off again, which shows that everything else is freely mixing. And so we could then just do the enzyme kinetics like Mo Cleland would have done back in '62, except we're now using a membrane enzyme in a detergent micelle, which is something that is not very often done in enzyme kinetics. Typically, people work with soluble enzymes. This is an enzyme that requires a detergent, and its lipids require detergent. But we got actually hyperbolic kinetics at a particularly useful detergent concentration. And we were able to show by seeing intersecting lines on these double reciprocal plots that this was a sequential mechanism and not a ping pong mechanism. And those of you who haven't taken biochemistry might not know what that means, but it basically means that there's two different kind of mechanisms where you could form a covalent complex with the enzyme, or one where there's a direct transfer of the group from one substrate to the other. And it turns out to be of the latter kind, known as the sequential mechanism, that's characterized by these intersecting lines down here. If they were parallel lines, then we would have to assume there's a certain kind of mechanism called a ping pong mechanism instead. And so we fit the data to the best equation, and it fit ordered by. But as a subsequent student did an experiment with a new lipid, lipid A instead of KDO2 lipid A, and they showed that they would get this sort of sigmoidal behavior. And now that's something that an iso mechanism can accommodate, but the ordered by mechanism cannot. So we were able to kind of figure out that PagP is behaving like an iso mechanism with the sigmoidal kinetics of a monomer. This can't be using some equilibrium model like Koshland-Nemeth-Filmer or the Monod, Wyman, Changeau models for allosteric control. That's not going to explain this because this is a monomer. Those things require oligomerization of subunits. So I think we have the best mechanism is an isotherel chance mechanism. And because it's an iso mechanism, we need to be able to see how does that fit into the catalytic mechanism if ever we propose one. And I will do so before the end of this talk. And you'll see exactly how the iso mechanism has to fit. And if it wasn't an iso mechanism, it would have to fit with this equation, but we can expect that under certain conditions with isotherel chance. It can accommodate the ordered by behavior, but it can also accommodate the sigmoidal behavior if, say, we're working at a steady state as opposed to a rapid equilibrium environment. So it takes a little bit of explaining to get the kinetics sorted out, and it's hard to do that in a brief talk like this, but it should give you a feeling for why this enzyme is kind of like a permease. So if you have a protein in the membrane that transfers groups from one side of the membrane by binding to them, and then it isomerizes to a form that allows the groups to come off on the other side of the membrane. Every reaction has to then be accommodated by the isomerization back to the initial state. So there's a conformational change that goes on in the protein cycle of transport. And PagP is doing that by transferring a palmital group from a phospholipid to another phospholipid. And it's acting like a permease itself, except it's acting not across the membrane, but between lipid phases within the membrane. So this outer membrane model here shows how the LPS is all on one leaflet and phospholipids are in the inner leaflet of the outer membrane. But there are mechanisms where too many phospholipids find their way to the outer membrane and they start accumulating in the external leaflet. And so there's a mechanism for vacuuming them up and transporting them back to the inner membrane to help maintain the asymmetry of the outer membrane. But if there is a situation where phospholipids are present in the external leaflet, PagP is there to use them to catalyze transfer of palmitate to lipid A. And it sort of will do so along the borderline of these clusters of LPS in the outer membrane. And so PagP is kind of acting when the phases between the lipids separate from each other, and it has to then make these separated phases create boundaries where there's dislocations in the membrane. We call them phase boundary dislocations, and they are a danger to the permeability of the cell. So when these sort of mutants involve phospholipid excess in the external leaflet of the outer membrane, they trigger PagP and they cause permeability to all kinds of antibiotics and detergents. And PagP is probably sealing up the boundaries to try and keep the permeability of the outer membrane together. So it's acting, it's produced in the outer membrane, but when the outer membrane has its lipid asymmetry intact, PagP doesn't do anything because it doesn't have any phospholipid donor to donate a acyl chain to the lipid A. But when the phases separate, when there's an imbalance in the correct amount of phospholipids, PagP is now able to take those phospholipids and use them to help restore the asymmetry of the outer membrane. So there are mechanisms for transporting lipopolysaccharide to the outer membrane that's driven by ATP binding and hydrolysis. There are mechanisms for transporting phospholipids back to the inner membrane, and that involves ATP binding and hydrolysis. There are also mechanisms for reacylating lysophospholipids generated in the outer membrane so that they become diacyl phospholipids again. And those mechanisms are also using acyl-CoAs, which are high energy forms of metabolism for driving these reactions. So the outer membrane is disconnected from all of the energetics of the cell. There's no transmembrane electrochemical potential or proton motive force to drive metabolism in the outer membrane. It's all in the inner membrane. But the lipids in the outer membrane are in flux, and they're driven by components that are connected to the inner membrane. So there is a steady state flow of lipids in the outer membrane. And we'll see how that has importance in seeing that PagP is actually functioning as a biosynthetic enzyme in the outer membrane where there's no energy source available, but it's actually energized so that it drives itself in the forward direction. And it's like a transporter of kinds, like a permease or a pump that pumps things across the membrane. PagP can pump them from one lipid phase to the other laterally. It's kind of neat. So our first insight into understanding it came from doing the Nmr structure in collaboration with Lewis Cabe. This was about 20 years ago now, and it was one of the first membrane protein structures to be solved in Canada at the time. And we used Nmr and found that there was this loop L1 that connects the first and second beta strands was highly dynamic. And it's to give a sense for what that is, it's just that the position of the amino acid residues in the side chain was all over the map, whereas the beta strands in the beta barrel of the membrane are very closely packed together, and they're highly ordered. The L1 loop is highly unstructured. And we think when we add a detergent that supports enzymatic activity, there's a transient state that is takes this L1 loop and organizes it over top. So it becomes structured and not disordered as shown. But the challenge with having figured out this enzyme and how it works, which I haven't explained to you how it works yet, but in coming to grips with it myself, the biggest challenge was that every structure we had is of a ground state structure, which only binds phospholipid but doesn't bind lipid A. And it was only through a simulation and through NMR that we could know there was an ordered state that was what we call the activated complex, which allows catalysis to occur. But it was a transient state that didn't show up in the structures, either by crystallography or NMR. But NMR could predict that there was a transient state. We called it the R state, which is relaxed. That's the ground state, actually. And then the T state is the ordered state. And the thermodynamic parameters that were assigned to this using what's called relaxation, dispersion, NMR, just gives you a sense for the fact that there is an organization in the activated complex that's compatible to catalysis, but we just can't see it because it's such a transient thing. It only exists for a fleeting time. So we had to kind of come to grips with it gradually. So the two things that we see at the beginning from our crystal structure in what's called lauryl dimethylamine N oxide, and we also got a crystal structure a few years later in sodium dodecyl sulfate and a precipitant that crystallographers like to use to grow crystals called methyl pentane diol. And we found from the two different structures that there was a closed and an open state of the protein. So this tyrosine here, tyrosine 147, forms an bidentate hydrogen bond to the backbone amide of leucine 125 here. So it's like a gate that closes a door. And in another structure where we have the SDS bound, this is LDAO bound here, the SDS structure included the tyrosine in a different conformation. It was flipped up, and so the door was open on the side. And we now know that this lateral gating is what gives the enzyme the ability to select a palmitate chain at the SN1 position. So this is a little simulation that shows kind of what happens. So the SN1 palmitate chain here knocks on the door, pushes the tyrosine out of the way, and dips its toe into the wall of the beta barrel wall of the protein. And so there's a lateral diffusion of the substrate into the interior of the protein. And we know that there's an ability of the enzyme to use palmitate at the SN1 position. And so if you, in black here is a phospholipid that has palmitate at the SN1 position. 16-0 stands for palmitate, and 14-0, that's for myristate. So this is a synthetic phospholipid which has myristate at the SN2 position and palmitate at the SN1 position, and that's the preferred substrate for the wild type enzyme. But if you switch the order so that the acyl chains are myristate at the SN1 position and palmitate at the SN2 position, the enzyme doesn't use that substrate very well at all. It's because it has a preference for palmitate at the SN1 position. Now, we were able to engineer some enzymes by taking advantage of a glycine residue at the floor of the, so the pocket where the acyl chain binds has a glycine on the floor. And if we add residue, larger side chains to the amino acid at that position, we can plug up the floor, and then the enzyme actually starts using shorter acyl chains. So that's how we identified what we call the hydrocarbon ruler. It's the pocket where the acyl chain binds, and we can make the dimensions of that pocket a little shallower, and now the enzyme uses myristate as its preferred substrate. And this one here, gly-88 serine, does that. And now it's using myristate at the SN1 position and not palmitate at the SN2 position. So we know the enzyme is Regio specific, and we know that the hydrocarbon ruler sets the chain to be fitting a palmitate and not a myristate. That's the wild type position. But we also noted that if we mutate that tyrosine 147, so it has a phenolic group that can hydrogen bond to the backbone amide. If we mutate it to a phenylalanine, we take the hydroxyl group off, and now we're basically designing the enzyme so that it can't form that bidentate hydrogen bond anymore. So it's going to be constantly in the open state because it can't be latched back to the closed state. And in these mutants, we find that they actually go a lot faster than the wild type. Their specific activity is a little more than double that of the wild type enzyme, but they have no regio specificity at all. They use palmitate only, and they don't care if it's at the SN1 or the SN2 position, unlike the wild type. And so we think this lateral gating of getting the palmitate to go in there is forcing the enzyme to use the SN1 chain. And we've often wondered, why would it want to do that? Well, if you transform bacteria with the mutant enzymes so that they're now produced in membranes, so before we were looking at purified enzymes working with lipids in detergent micelles, now we're looking at biological membranes from cells using those mutants. We were able to show that these mutants start putting stearate, which is a C18 acyl chain, into their lipid A. And we were surprised by that. One of the myristyl transferase mutants we made still puts 14 carbon acyl chains in, but these ones were able to add 18. And we didn't really know how to explain that, except that we think that the palmitate chain, if it gets in there, it's at a position that lines up to create the transition state in catalysis, which I'll summarize for you momentarily. But if a myristate chain goes in, it's too shallow to be able to react, so it gets rejected. But if a stearate chain goes in, it's a bit too long, but because there's room for it to kind of distort its backbone, what we call gauche conformers, it can actually fit itself in quite nicely. And so if stearate is getting into these mutants, as the evidence shows, that means that there must be some kind of mechanism for keeping stearate out, because we've never seen it in lipid A in wild type cells before using PagP. So we think that what's going on is that the preference for the SN1 palmitate chain is to exclude stearate, which is primarily found at the SN2 position. But if you have a mutation that takes the enzyme's ability to select the SN1 chain away, now the stearate chains can be incorporated. And so probably natural selection is acting on making the enzyme so that it's specific for palmitate by using two different approaches combining together. One is to have regio specificity to keep the SN2 chain out, because that's where the lion's share of the stearate chains are esterified in the E. coli phospholipid pool. The main acyl chains to deal with at the SN1 position are palmitate and myristate, and the hydrocarbon ruler appears to be able to take care of the myristate by rejecting those acyl chains. So if the SN1 chain presents myristate, that's going to get rejected. If the SN1 chain presents palmitate, that's accepted, and it will drive the isomerization to the activated state that allows the enzyme to catalyze the reaction. So we're getting to the point where we can explain how the enzyme is specific for palmitate, why it doesn't select a shorter acyl chain or a longer acyl chain, which are both present in the phospholipid pool. And so we can explain the specificity, but we still don't exactly know why palmitate is so important. Maybe it's because of the way that acyl chain interacts with the receptor for lipid A in the human immune system. Perhaps that's the explanation. So we've been able to kind of walk along the hydrocarbon ruler. So this is the glycine 88 at the floor here. And palmitate chain has 16 carbons and would come down to here. And we've engineered the floor with an alanine, a cysteine, and then we use the cysteine and chemical alkylating reagents to modify the cysteine so it can look a lot more like an acyl chain itself. And as we plug up the floor of the hydrocarbon ruler, the enzyme starts being selective for 12 carbon and 10 carbon acyl chains. But after that, the logic can't be extended any further because there's no more binding. There's nothing to contact through van der Waals interactions in this little hydrophobic pocket. So this is probably why the enzyme works the way it does back here. It's only holding on to the bottom of the acyl chain, and there's all this space in here where it's able to distort itself. We can't actually keep stearate out unless we keep the SN2 chains out using this Regio specificity. So there's Regio specific selection of palmitate at the SN1 position, and that makes the enzyme work the way it does. These are some examples of how phospholipids come up close to the outside of the beta barrel and make contact, and will have to push their way through by pushing the tyrosine out of the way. And when it comes up, then the acyl chain can move in and bind inside the hydrocarbon ruler. So we've been asking questions about how do acyl chains get through the wall of the beta barrel. There's prolines that are amino acids in proteins that are unusual in that they don't have a backbone amide to donate a hydrogen bond in the beta strands that form. And so they actually create little discontinuities of hydrogen bonding between the beta strands. And so Pro28 and Pro50 are close together, and you can't really see it in sticks format, but if you showed it in a van der Waals surface, there's no space between these residues. There's just a direct contact between them. And so you can't really see what's going on the other side of the beta barrel wall. And you can see there's a detergent bound in there because we're showing it in the sticks format. But there's like a little opening in the wall that allows something to get in there. If those prolines could separate, you would be able to see inside. And in fact, this is part of the mechanism that the enzyme goes from having a closed wall so that the acceptor substrate can't get in to one that opens up and allows the substrate in. So how do we know that? Well, we mutated these prolines to cysteines, and then we used a hydrophobic oxidizing reagent to drive formation of disulfide bonds. And when we did that, we ended up getting enzyme that was stable and still had phospholipase activity. It's a promiscuous activity of the enzyme in my cells. About 20% of its activity is a phospholipase. That activity was not affected, but the ability to transfer palmitate to lipid A was lost. And the opening where the phospholipid goes in is on the other side of the enzyme. So there's two openings, and we call one of them the crenel. It's like this turret of a medieval castle. It's a little indentation in the wall. That's basically what this is in a beta barrel, and we call it the crenel. It's where the phospholipid binds, and in the previous slide, the opening where the lipid A binds is called the embrasure. And it's more like a window rather than a notch, as we see over here. And we have spectroscopic and chemical evidence to confirm that these modifications worked. And under the conditions where we have phospholipase activity, we can lose the ability to transfer acyl chains to lipid A, which is a lateral acyl transfer process, rather than allowing a water molecule for a hydrolysis. That activity is blocked when you have oxidation of the two prolines where they've been substituted with cysteines. And so the disulfide bond stops the activity, and if we then reduce it again to open things up, we get all our activity back. So it was very convincing that we need to open the wall of the beta barrel to allow the acceptor substrate to go in between Pro50 and Pro28. So it's going to come in through the side, and the phospholipid goes in through this gating process that involves throwing this tyrosine into a different position. There's also a couple of prolines in there as well, and they probably contribute to the opening as much as opening the tyrosine. So I think I said a lot, and probably if I just run a little simulation of what we think the enzyme's doing in membranes, this is a model where the phospholipid binds at the crenel. It dips its toe into the hydrocarbon ruler. There's a couple of aromatic side chains that have spectroscopic properties we know about. And there's an arginine that electrostatically interacts with the phosphate head group. But these two prolines, proline 50 and 28, they need to come apart in order for the acceptor substrate to get in there. And We also know that the two residues involved in catalysis, histidine 33 and serine 77, are too far apart from each other to be able to react. So we figured out how the palmitate gets in through the crenel by this lateral gating. The hydrocarbon ruler makes sure only palmitate will be accepted, but we still can't explain how the enzyme catalyzes the chemical reaction. So we looked at a molecular dynamics simulation like that, and we asked the question, are there any transient states in the simulation where we can see the separation of the prolines? And in fact, there is. It only happens a fraction of the time, and the opening in the wall is sufficient probably for a lipid A to project its hydroxyl group as an acceptor for the reaction. So this is the work that Mo Shariari did. And what we've done is we've compared the ground state structure, which is shown here on the left in side view and on the right in top view, where the his and the serine are too far apart from each other, and where the prolines are close to each other. And we've then taken the transient excited state where the histidine and serine come close together and the prolines become separated. And we'll run a little morph of the transition between those two states. And the spheres will disappear. So now the prolines here are going to come apart, and the histidine and the serine are going to come into close proximity over here. And so that's where we're going to see the catalytic state, where the histidine is interacting with the serine and the prolines are separated so that the acceptor substrate can let itself in. And after the reaction occurs, then everything relaxes back to the ground state where the histidine and the serine are now far apart. So it's important to know at this state that the L1 loop where this histidine is highly dynamic. And so this is an environment that's solvent exposed. But when the histidine and the serine come into close proximity and the L1 loop orders itself at the top here, that state is a solvent shielded environment. And so that helps keep water out of the reactions to promote the acyltransferase reaction. So that's important to understand because part of the mechanism involves changing the structure of the imidazole in a subtle way that needs to be accounted for. So we'll just show one more time. The histidine and the serine come close together. This is the isomerization to the activated complex after the acyl chain at the SN1 position is confirmed to be palmitate. This new conformation forms. We actually have spectroscopic evidence to show that it exists in addition to the NMR data. But the relaxation of the enzyme back to the ground state, this is a macromolecular conformational isomerization, and it happens on a slow time scale compared to many enzymes that catalyze very rapid chemical reactions. It happens kind of like a translocase transporting substrates across the membrane. It's on that kind of conformational time scale that the enzymatic reaction is actually dependent on the isomerization of this stable enzyme form. So let me explain to you why this is important. So if you look at the enzyme from a cross section like this, you can see there's no real access between the hydrocarbon ruler and the exterior lipids that are in the membrane. And so we need to have these lateral gating processes. And so this is one crystal structure. This is the one with SDS where you can't see how the lipid A acceptor would get in there. But after the L1 loop, which is dynamic in the ground state, becomes ordered and above in the activated complex, it creates a little opening in the beta barrel wall. You can actually see some light through there. And that's probably where the lipid A acceptor has to go. So in the ground state, the histidine and serine are far apart, and the proline 50 and pro 28 are close together. But in the activated complex, the his and ser come close together and the prolines separate. And so if we look at the histidine, its atom called N delta 1 is pointing down. That's important. And the contact between it and the serine is actually through C epsilon 1. So that's unusual for a histidine and a serine to interact through the carbon of the histidine rather than one of the nitrogens. Usually in a serine protease, it's the nitrogens that interact with the serine. And so they can act as a base to abstract a proton from the serine, making it nucleophilic, and it can react. In this case, the serine doesn't become reactive. It just acts like a hydrogen bond donor. And the histidine does all the chemistry, but it's interacting with the serine through that C epsilon 1, which is kind of interesting. So his33 N delta 1 is pointing down. You can see again that it's right under that opening. So when the lipid A eventually comes in to go through that opening, the phospholipid's already bound at the crenel. The serine and the histidine are, in this case, they're close together. You can't actually see their alignment because I've turned the axis on in a way that kind of highlights the prolines instead. But the prolines are close together, and there's nothing that can get in there. When the prolines are far apart, there's an opening, a window in the embrasure, which allows the lipid A to insert its hydroxyl group. And so this is the SN2 carbonyl of the phospholipid acyl chain that dangles on the outside of the crenel. And the one that's in the hydrocarbon ruler is here. And it's going to move a little closer to the serine. And the histidine and the serine and the carbonyl oxygen and the hydroxyl group of the lipid A acceptor will all come into close proximity. You can see it again here from the top view. The histidine and the serine are far apart to begin with, and then they're close together. And once we get started, I think what's going to happen is we'll have this imidazole side chain of the histidine can exist in two conformational states. One where the nitrogen is has a lone pair of electrons, so that's the basic form, and the N epsilon nitrogen is protonated. And so this is the common form of imidazole that's called the tau tautomer. But you can actually flip it around so that N delta 1 is protonated and N epsilon 2 is in the basic form. And we're actually going to do that. So you'll see how it goes. So if the hydroxyl group of lipid A is presented into this opening in the beta barrel wall at the embrasure that comes from the spreading out of those two prolines, and the histidine is contacting the serine through C epsilon 1 like this, we can imagine the N delta 1 nitrogen acting as a general base to abstract that proton. And that will cause the electron to go to the oxygen, and that will attack the carbonyl carbon, and we end up forming a tetrahedral transition state. And so the negative charge in the transition state is the main culprit in its instability. And so by the serine providing its hydroxyl group as a hydrogen bond to stabilize that, you can also get some electrostatic stabilization, presumably from the positive charge of the imidazole, which has now become an imidazolium cation. But this is actually a pretty crummy way of directing that positive charge. So electrostatic stabilization, I put a question mark here, but I can easily imagine if this ring flips by 180 degrees around its dihedral angle known as chi2, it will bring the N epsilon 2 nitrogen into proximity instead of the C epsilon 1. And that's a much better way of stabilizing the transition state. In fact, it might lead to some kind of low barrier hydrogen bond. And we're now in a position where we can do general acid catalysis and protonate the leaving group after having flipped that imidazole. So the mechanism suggests that there is a slow rotamer rotation of the imidazole orientation after it abstracts the proton in order to deliver the proton to the leaving group. And so in the end, we end up with a palmitoylated lipid A and now a new alcohol. So we started out with an alcohol and an ester, and we ended with an ester and an alcohol. But that's the free energy of that reaction has a delta G about 0. And so the equilibrium constant is equal to 1. And so this reaction is just an equilibration. However, we now have to consider that the imidazolium side chain is now been isomerized. And now we have this pi tautomer that's formed, which has N epsilon 2 as the lone pair of electrons and N delta 1 with the proton. That's not how the reaction starts. And the enzyme isn't finished yet. So we have to do something. Well, what's going to happen is the enzyme will now move back to its ground state. And before that can happen, the palmitylated lipid A has to be released. But when the imidazole moves into the dynamic environment that we showed earlier, where it's exposed to water, now the water can be used to dissociate the proton at N delta 1 and to reprotonate N epsilon 2. And this actually occurs spontaneously. And the equilibrium lies far to the right. So it causes the reaction to become effectively irreversible. And so by the enzyme sampling these hydrophobic and solvent exposed hydrophilic environments through the conformational change, it can actually drive the reaction forward, kind of like a molecular turnstile. So every time the reaction occurs, it only goes in one direction. And it's acting like a permease, but it's moving the acyl chain laterally from a phospholipid phase to a lipopolysaccharide phase. And it's doing it at the interface. And so it's kind of neat. It explains how the enzyme works. So I kind of drew a little blue circle here to show the opening in the embrasure wall to get a sense for putting it all back together again. First, the hydroxyl group of the lipid A acceptor goes in through the opening, and the N delta 1 of the imidazole abstracts that proton so you can form the tetrahedral intermediate. And then the imidazole has to flip its ring. And I didn't show you in the previous slide, but there's an asparagine on the other side, which kind of can accommodate this flipping very readily. And so that allows the leaving group to be protonated by the reverse of the acylation, except it takes a new path now because of the flipping. And that kind of drives the reaction forward as well. It contributes, it prevents the reversibility of the reaction, basically. The flipping serves to drive the reaction forward. So the free energy of the reaction is 0, but the tautomerization of the imidazole back to the tau state is thermodynamically favorable. So that drives it's part of the reaction. And so that is the step, this tautomerization of the imidazole at the end, after the final product's been released, that explains the iso mechanism. So that's what this E prime to E is at the end. It's new terms in the rate equation have to be added whenever you have an iso mechanism. But you can also simplify the mechanism with the Thorell chance, where you take out the lifetime of the ternary complex, and then you end up with an equation that algebraically is identical to ordered by, which is something that can't accommodate any of this, the sigmoidal behavior we were seeing with lipid A. So I think the mechanism is due to the fact that this is a membrane protein, and the membrane is an energetically, it's the energy transducing part of the cell, especially the cytoplasmic membrane, but this is happening in the outer membrane where there are no conventional sources of energy. But as I kind of illustrated earlier, the ATP binding cassette transporters and acyl-CoA in the cytoplasm, which is the conventional energy sources of the cell, they are being used to drive processes that are transported to the outer membrane. And so the lipids in the outer membrane themselves are in a steady state flux, driven from the inner membrane. And PagP is a membrane protein in the outer membrane, and it can basically pick up the lipid with the right acyl chain and attach it to lipid A. And it can do that as a biosynthetic enzyme in that external extracellular environment. And so it gives us some new insight into how enzymes function in biological membranes. So there's two major phases where the kernel is closed and then open, and then the phospholipid is interrogated. The hydrocarbon ruler will reject acyl chains that are too short, But if palmitate is found, that will be recognized and that will drive the isomerization to the activated complex. That involves the prolines coming apart and the histidine and serine coming close together. And that's a different conformational change. So there's two conformational changes. The one that's I call induced fit, that selects the palmitate, and the isomerization is forming the activated complex that allows the catalytic process to proceed. So it took only 20 some years to figure that out, but that's part of what we've done so far. And I wanted to share that with you. I think I'm basically at the end of my time now. And I put that together on short notice. Mo Shariari made the morphomation of the activated complex, which we've just done recently. It came out of the work on the molecular dynamics simulations that was done with Chris Neal, now at the Los Alamos National Laboratory, but he did his PhD with Regio. We're still working with Regio now, looking at this, and the crystallographic work that we did with Gil Prive helped immensely, and the NMR work with Peter and Lewis was also essential. And the enzymology was done and the site-directed mutagenesis by graduate students working in my lab. And I'm glad I had a chance to talk to you. I know nobody asked any questions. Maybe I just speak too fast, but we can try now if you have time.
Maybe if anyone wants to unmute themselves, they can to ask a question. If not, they can enter it into the chat.
Should I take my screen sharing down?
If they feel more comfortable to share their
No, I meant for me
cameras.
to clear the screen so that we can see people's faces.
It doesn't look like anyone is showing their faces, but maybe if we need to go back to a slide, we might need the screen share.
Okay, I'll leave it like this.
While we're waiting, I do have a question of my own.
Okay.
I'll get us started to break the ice, I guess. When you were talking about the hydrocarbon ruler for PagP, if you were to mutate the hydrocarbon ruler to allow for stearate, would the reaction still, like, would the histidine and the, would it still close for the PagP reaction?
Well, so the hydrocarbon ruler, that's a good question you're asking, and you'll see why it won't work once you think through it a bit. But the glycine is the amino acid which doesn't have a side chain, right? Just has a proton. And so glycine is the shortest acyl chain you can get, and it's lining the floor of the hydrocarbon ruler. And the hydrocarbon ruler is designed for a 16 carbon acyl chain. So you can fill it up and plug it up, and you can make shorter acyl chains get selected, but you can't make longer acyl chains get selected by modulating the hydrocarbon ruler, because you can't make an acyl chain smaller than glycine. So you can mutate glycine to alanine, and that enzyme will now use a 15 carbon acyl chain. If you mutate glycine to so say valine, it's now starts using a 14 carbon acyl chain. But you can't make it, mutate it to use a longer acyl chain, because you can't make it any longer. You can't make the pocket any deeper. It's already limited by the structure of the protein. So how did I get 20 in? Well, that's because we messed with the Regio selectivity. So the reason that 18 carbon stearate chain, I should get that straight. The 18 carbon stearate chain went in because the Regio specificity had been affected. And that came not from mutating the hydrocarbon ruler, but by mutating the tyrosine 147 that was gating the crenal. Remember, I mutated tyrosine to phenylalanine. And so you can get longer acyl chains to be used by the hydrocarbon ruler, but not by changing the hydrocarbon ruler. You do that by changing the lateral gating through the crenal.
Oh, but then would it not be able to then proceed with the reaction since the crenal was mutated at that point?
So let me think. Okay, so the reason that stearate, C18 stearate gets in, it was an unexpected result, okay? We didn't think the enzyme could use something longer than palmitate, but when we made the crenal gating mutants, where we mutated tyrosine 147 to phenylalanine. That's a very subtle substitution, mind you. You know, like phenylalanine and tyrosine differ by a hydroxyl group. It's really a small difference. That mutation created an enzyme that completely lost its ability to have Regio specificity. And unexpectedly, it started using stearate. Well, we think that's because stearate is one of the main phospholipids at the SN2 position, and the enzyme is specific for SN1 in order to keep the stearate out, because we never seen stearate before, but now we know how it gets in. It gets in by crenal gating, by if you defeat that, if you mutate it so it doesn't work anymore, then the enzyme starts using stearate. You can make the enzyme use shorter acyl chains by mutating the hydrocarbon ruler. So it's the two things combined, given that the phospholipid pool contains basically a combinatorial library of acyl chains of 14, 16, and 18 carbon lengths that are at the SN1 and SN2 positions. The enzyme can pick palmitate out of that. And it goes to a lot of trouble to do it. And now, we didn't know how to explain how any enzyme could do that. Not many enzymes do that, but still PagP does it. And we now can explain how it happens.
That makes sense.
Lateral gating for regiospecificity plus hydrocarbon ruler measurement to exclude the myristate. So you exclude the myristate at the SN1 position with the hydrocarbon ruler. You exclude the stearate by making it regiospecific for SN1 because all the stearates at SN2. Does it make sense?
That makes sense. Yeah, you're kind of acting with two filters so that you can get.
That's right. That's exactly it. You get it. Okay. It's basically a double sieve.
Yep. Well, I guess no one really has any questions. The lecture must have been amazing and clear to everyone. So thank you again, Dr.
I wish I would have had
Bishop.
a chance to find out what the background of the people watching is, because it's really hard for a guy like me to gauge my audience. What if you're all a bunch of whiz kids and stuff? I want to give you the stuff that maybe I should have eased you into it more slowly. What do you think? Did I do okay? I
enjoyed the lecture personally. I'm sure everyone else did also. I think that
you've taken my class, so you have an advantage.
I do have a slight advantage there. Seeing that there doesn't seem to be any questions, I guess everyone understood everything perfectly. So if you do think of a question, I will drop your email if that is okay.
Sure. And thanks for inviting me to do this. I hope it wasn't a complete waste of time for you.
It was not. My keyboard is not working. So, Dr. Bishop, if you could drop your email into the chat for everyone. I do not know why my keyboard has stopped working.
Live
but I'll
transcript, record, chat. There's chat. So
I'll thank everyone for attending. It is a little bit of a smaller crowd, and we seem to have dwindled a little bit even more during the course of the lecture. It seems like people couldn't handle the sheer wealth and depth of knowledge that you were giving us. So I thank everyone again, and I thank Dr. Bishop for giving this amazing lecture. We do have another lecture on Thursday given by Dr. Giuseppe Malencini, who will be giving a lecture on cytokines, I'm pretty sure. So stay tuned for that. You should have gotten an email for that. If it's free to attend to anyone. If you have friends that might be interested, send them an email for that. And thank you again.
Giuseppe will teach you a lot about NMR, I'm sure.
Oh, we have a question from the chat asking about what regiospecificity is.
What is regiospecificity? So the phospholipid has two pieces of spaghetti attached to it. One is at the SN1 position and the other is at the SN2 position. Those positions are sometimes enzymes can't really distinguish one from the other. It'll take an acyl chain from a phospholipid from either position and transfer it. Those ones would not be regiospecific. PagP is highly regiospecific in that it only acts on the SN1 acyl chain. Now, if we mutate tyrosine 147 to phenylalanine, it starts using both. So it loses its regiospecificity.